Alright everyone, welcome back to Marrat Academy. I am Yasser Bashir, and today we will be discussing this paper 6 paper, Alternative to Practical, October-November 2025, Variant 2. This is a 16-page paper, including 8 like pages, and let's get right into it. So here we have question number 1, of course, it's the classic multi-step investigation question. So let's just read it step by step and understand what's really going on. A student investigated the rate of respiration in yeast using different concentrations of glucose. They used a 5% glucose solution to make a 1% glucose solution and a 4% glucose solution. Okay, the student used this method. First of all, we have to have two separate test tubes, one of which we will label as 1%, and the other we will label it as 4%. Now, here we want to prepare both the 1% and the 4% one. For the 1% test tube, the way that we will prepare that is that we will have this 5% glucose solution, which is the source. It is the stock solution. And a stock solution just acts as basically the source in which we will use to dilute any subsequent concentration. So what we'll be doing here for the 1% is that we'll simply just take 1 centimeter cubed using a syringe into the 1% test tube. So here we'll just fill it with 1% or like 1 centimeter cubed of 5% glucose solution. Now, on the other hand, when we talk about the 4% one, here, instead of actually filling it with 1 centimeter cubed of glucose, we will instead use 4%. Now you might be asking me, okay, so after all, this is still 5%, this is still 5%, what's different really is just simply the volume. So technically, it's still the same concentration. You're correct. This is why we still have step 3. In step 3, what we'll be doing is that since this one is 1 centimeter cubed, and this one is 4 centimeter cubed, we want to make them both the same volume. You want them both to be 5 centimeter cubed. How do you do so? Basically, we will add for the 1 centimeter cubed solution, we will add 4 centimeter cubed of what? Of distilled water, which is absolutely pure water, pure H2O with no chlorine or anything. And then for the 4% test tube, then here you add 1 centimeter cubed of water, of distilled water, so that the total of both of them will be 5 centimeter cubed. And this will of course make the 1% test tube as 1% glucose and the 4% test tube as 4% glucose. So this is how we have prepared them all. Okay, so let's see what we have here for step 4. It says that we should pour yeast suspension into the small test tube labeled 1% until it's completely full. Okay, so essentially what we are doing here is that for the 1%, if you see in the illustration here, when we've added the 4% centimeter cubed of water, making the whole thing a total of 5%, or I mean 5 centimeter cubed, you notice that there is still a gap here. So to fill this gap, what we'll be doing is that we will be adding till the very top, we'll be adding yeast suspension. Okay, so any gap that we have here, we will fill it with what? With yeast suspension. Because after all, this investigation is about what? It's about the rate of respiration of yeast. So of course, we have to have yeast in our experiment. Now the same goes of course for the 4% one, but that will be later, not now. For now, we only focus about the 1% yeast solution. Or to be more specific, the glucose solution. So what we do with that now? Okay, now we have a fully filled test tube with glucose, distilled water, which of course makes it 1% glucose. And then we fill that with a yeast solution. So that it's filled to the very top. What will we do? So basically here, we already have this test tube, right? What we'll be doing here now is that we take another test tube that is large and we invert it on top of the test tube. So basically here, we have the large test tube, right? We just suspend that maybe we use a clamp or whatever. We make sure that it's secure. And then our small test tube, we insert it very gently, making sure that there is no solution that would spill whatsoever to ensure that it is completely sealed. So basically here, we put the small test tube into the inverted larger test tube and make sure that it completely touches that test tube to make sure that it's completely sealed. Once that is sealed and once that's figured out, then essentially what we'll be doing is that in a swift and in a fast motion, we just flip the test tubes. So this case, or like when we do that, this would make the larger test tube upright, while the smaller test tube, it would be inverted. It would be like so. Now after we've got this shape here, after we have this test tube, the smaller one being inverted, what we'll be doing is that we will be inserting this setup here into a water bath. This water bath will contain warm water. This is of course to ensure that temperature has no effect on the experiment, because after all, we are testing on the different concentrations of glucose. So if the temperature did vary, then this may affect the experiment as well as the results. So we don't want that. We want to make sure that any result we get is only because of the concentration. Okay, now after we do that, we will start a stop clock, or a stopwatch if you may call it, for about 5 minutes, and then we observe. We observe the height of the yeast suspension that leaked to the bigger test tube. And the way that we assess the amount that got leaked out, we use a ruler. We have a ruler here. You make sure that it is basically upright and parallel. And then you figure out and measure this length here. Okay, so we got this length over here. So what do we do next? Basically, we remove the test tube and the warm water and completely discard everything. We start from scratch and do the same procedure over here for the 4% solution. Because remember, this whole procedure here was only for the 1% solution. So what we want to do now is that we want to do the same exact thing for the 4% solution, and then we will do the same experiment and get a different reading for what? For this height over here. And then we can take note of that and convert it into a table, which is exactly what question A1 asked us to do. Essentially what we have here is that we have the illustration for the 1% glucose solution compared to the 4% glucose solution. Now after that, what we'll be doing is that essentially we will be using our ruler in the exam, our actual physical ruler, and then we make sure that we read from the bottom of the meniscus. Make sure that you read from the bottom of the meniscus and not from this part here. You don't read from here. You read from the bottom, which would be here. So the best approach is to draw lines like so, like that, and then using your ruler, your plastic ruler or whatever, make sure that it is like at least transparent. That would be easier to assess. And then you can actually write the values here. Now this is not part of the question. You don't write it here literally. However, you just write it here as a draft. And then from here, what you can do is that you can draw the table. So essentially, we want to figure out the correct format of the table that we have. First of all, we have two categories. We have the 1% glucose solution and we have the 4% glucose solution. And we are comparing that against the height of the yeast suspension in the larger test tube. So really, we only have two columns that we need to be concerned about. So just for the sake of making it easier for us, I'll make this table a little bit wider, and then I will split that into two columns, like so. And of course, make sure that you have a row for the header that includes the units. So essentially here, what I will write here is the glucose solution concentration in test tube. And then of course I will write the unit. So, because of course, after all, we don't really have labels. We didn't label that one, we didn't label that A, and didn't label that B. We just identified that as 1% and 4%. So, that is exactly what we'll be doing here. We will write here percentage concentration of glucose solution in test tube. Okay, that sounds pretty good now. However, you have two ways of coming around this. You either write it as I did, or you can just write concentration of glucose solution in test tube and then slash percent. And you completely omit this percentage here. But in my case, I did it like so. Now, this is the first heading done. Now we want to go to the second heading. So we have the height that we want to assess. So the height here, this height, supposedly this height, the height of the used solution in the larger test tube. That would be in centimeters probably. Centimeters or millimeters, either. It really depends on you. However, it is always better to use millimeters, because in biology, we rely more on millimeters. So that's essentially what we'll be doing here. We will be writing the height or length, or I'll just say height. So that is basically what I'll be doing here. So I will write here height of heat suspension or solution, to be more specific, in larger test tube. And then here you have to make sure that you write the unit. And the unit should be millimeters, as we discussed. Now here, since we only have two values here, then you can just split that. So that'll be 1 and that'll be 4. You may be asking me, do I just write percent here? No, you should not. Because we already put that in the headings. We already put that unit in the headings. So you should not whatsoever put the unit in the table here. So instead, we'll just be writing 1 and 4, respectively. And then we actually measure using a ruler. Now, unfortunately, since I'm solving it digitally here, I don't really have access to a ruler. So instead, I'll be relying on the value from the marking scheme. And according to the marking scheme, this value over here for the 1% solution should be about 13 plus minus 1 millimeter, which basically means that the accepted range for this question over here is 12, 13, and 14. So I will write here 13. Now, of course, this depends on you and how you measured it. So you should use your own ruler, and it should be between this range. Now, maybe when you actually print out the paper, it won't exactly be in this range, because the marking scheme takes the measurement from the actual printed paper ready for the exam. So your paper that was printed at home may not have the same proportions. But as long as you did the measurement right, you should be good to go. And this one over here should be 23 plus minus 1 millimeter, which means that the accepted range will be 22, 23, and 24. And I will write here 23. Again, you do not write the units because you already did so in the heading. And this is what you should do. You cannot just erase this and write millimeter. You can't do that. It has to be in the headings. Otherwise, you will not be getting the full marks or full credit. Okay, now moving on. Here for part 2, it says us or asks us to state a conclusion for the results of this investigation. Essentially, what they want us to do is that we should either use illustration or the table, which is a bit more clear, to conclude a conclusion. So basically here, we can see that for the 4% solution, the height was greater. So this is exactly what we would say. We can say that the relationship between the percentage of glucose solution and the height of the yeast solution in the largest tube is directly proportional. Directly proportional means that if one increases, the other would increase. And this is exactly what happened here. So this is what I would say. So the relationship between the glucose concentration and the yeast solution, and the yeast solution, or the height of the yeast solution, height, if you want to be more specific, the height of the yeast solution is directly proportional, which means that if one increases, the other would respectively increase as well. Directly proportional. Now, this is actually a term that isn't only restricted to biology. It is a scientific term, which means that if you want to use it in physics and chemistry, you can as well. Though I'd say that it is most common in physics compared to the other two. Okay, now for part 3 here, state the independent and dependent variables in this investigation. Okay, now the independent variable is the one that you change, the one that you alter. And the dependent variable is the one that you observe and measure. So the thing that we have changed here is the concentration of the glucose solution. So this is exactly what we were right for the independent variable, concentration of glucose solution. Okay, now as we're done with that, we should go to the dependent variable, which is the one that we measured. Now, after we have altered the concentration, we have measured what? We have measured this length over here, the height. So the dependent variable will be the height of the yeast solution in the larger test tube. Height of yeast solution in the larger test tube. Okay. Now describe how the temperature of the water bath could be determined. Okay, or could be maintained actually. Maintained. So to maintain the water bath, because after all this is a warm water bath, as we have seen here. Yeah, this is a warm water bath. And if we just kept it as is, then after time it will gradually and exponentially become cooler after all. It will try to match the environmental temperature. So instead, what we're doing here is that we'll put this water bath in a thermostatically controlled water bath, which is basically an electronic device that would, by electricity, turn on a heater to heat the water to a certain degree. And this will always be constant. And this is something that you have to make sure that you memorize, as it will be guaranteed that it will come or be used to some degree in your actual exam. This is always a point that's used every single year in any paper 6 question, or to be more specific, any paper 6 paper whatsoever. So, what we'll be writing here is the use of a thermostatically controlled water bath via using a thermostatically controlled water bath. Okay, now explain why a clean syringe was used in step 3. So let's go back to step 3 and read what that is. So for step 3 here, use a clean syringe to about 4 centimeter cubes of distilled water into the small test tube labeled 1%. Then, put 1 centimeter cubed of distilled water into the small test tube labeled 4%. It has to be clean. So it's asking why should this syringe be clean? Essentially, if we used a syringe that was previously used for anything else, maybe with what a chemical in it, then essentially it will contaminate the solution and potentially alter the results. So because we don't want that to happen, what we'll be doing instead is that we will just be making sure that the solution is standardized using a clean syringe. So this is in order to avoid contamination of the solution. So to avoid contamination of the solution. Okay, that's good. I'm moving on. For part B, a student extended the investigation by using more concentrations of glucose solution. Calculate the volumes of 5% glucose solution and distilled water that are needed to make 5% of a 3% glucose solution. Okay, so here, even if you're not really sure about that, you can take the previous test tubes as a reference point. So, to make a 1% glucose solution, what we did is that we took 1 centimeter cubed of a glucose solution, and then we've added 4 centimeter cubed of what? Of distilled water, to make everything to add up to 5 centimeter cubed. So we want to ensure the same thing. We want to ensure that the overall total will be 5 centimeter cubed. So if you want a 3% glucose solution and we used 1 centimeter cubed to make 1%, then of course we can also use 3 centimeter cubed to make 3%. So the volume of 5% glucose solution, which after all is the source, will be 3 centimeter cubed. So we add 3 centimeter cubed of the 5% glucose solution. Then to add it up to 5%, then we should add a further 2 centimeter cubed of distilled water to make everything up to 5 centimeter cubed. This is how you assess and answer this question. Okay, sports drinks often contain glucose, which is a reducing sugar. State how you could test a sample of a sports drink for reducing sugar. Okay, alright. Now to answer this question, you have to be familiar with the tests, the biological tests. So for reducing sugars here, the test should be the Benedict solution test. Use the Benedict solution test. And then you should describe how you use it. Now here it says state. It doesn't want us to actually describe or explain how it should work. So you're okay with just saying that, like, you should use the Benedict solution test while heating. That will give you a full credit. However, if this question was instead a describe or explain question, then you have to be more detailed. So the extra points that you would be using in such question would be as follows. I'll be writing that in red to differentiate. Basically, yeah, you heat it, but you heat it till boiling. So use the Benedict solution test while heating until boiling. So that should be 100 degrees Celsius. And then you should actually look at the colors. Okay, so basically here, observe the color change. Observe the color change. And then you should basically describe what a positive result would be. So positive result, it would typically be brick red. For the Benedict test, it should be about brick red. That's if there was a lot of reducing sugar. If it was a moderate amount, it'd be yellow to orange about. Yeah. So yellow to orange, that'd be about a moderate amount. A intense concentration of reducing sugars will be about to break red, and absolutely nothing would be blue. So initially it's blue, and then it turns brick red if there's actually reducing sugar. Okay, so that's that. Let's go on with part D. The ethanol emulsion test can be used to show that a sample of food contains fat. State the result of a positive test. Okay, so they were generous enough to give you the test. So now you should recall what the positive test would be, the positive result. So when there is fat present and you use an ethanol emulsion test, then the solution will turn cloudy. So the result would be cloudy solution, cloudy or milky solution. Either works, up to you. Now for part 2, suggest one safety precaution that should be followed when using ethanol. Now ethanol is essentially a fuel. It is actually a biofuel. It can be used in petrol. It can be mixed with petrol. So because of that, of course, it is a safety hazard when it comes to fires. So you should ensure that ethanol is stayed away from heat, from any fire. Okay? So ensure ethanol is not exposed to fires. To fires. Okay? Now that should do it here. This is for question number 1. However, of course, we have question number 2 and question number 3 to do. So let's first of all start with question number 2. So for question number 2 here, plan an investigation to determine the effect of exercise intensity on heart rate in humans. Okay, now I want you to do one thing. Whenever you see any such question, then essentially what you should be doing is that you should always follow a template. Now the template that I always suggest my students to follow will be as follows. First of all, the list here, the independent variable in a neat way. So here you have the independent variable. And then here you have the dependent variable. Of course, both are followed by a colon. And then you have the constant variable. Here you try to basically mention as much variables as you need to or have to. And then you write the method. And then after all, you write the precautions, the safety precautions. Safety and precautions. Now you may choose to start from anywhere you would like, but personally I will start with the independent variable. Now for the independent variable, essentially what we are assessing here is the effects of exercise intensity on heart rate in humans. Now here you can assess that in different ways actually. Exercise intensity, it isn't only exercise. So you can assess that by like different methods. For example, what you can do is that you can, for example, test on a variety of exercises. Like for example, you can test running compared to walking, compared to like let's say hiking or whatever that is. You can do that. Or you can have one specific exercise, but to vary the intensity, then you vary the length and the duration of the exercise. Both are viable. So what I will do is that I will go with the latter, and I will essentially just say that we'll be using the same exercise, but we vary the duration of the exercise. So the independent variable is exercise. That's with a C. Exercise with various length. Various durations. Okay. Now with that out of the window, we can move on to the next part here, which is the dependent variable. Essentially for the dependent variable, it is what we measure. So we essentially want to measure the heart rate. This is exactly what the question says after all. So the dependent variable will just simply be the heart rate. Now we want constant variables. Now for the constant variable, since here we are actually varying the durations and not the exercise itself should be constant. So after all, the constant variable in this case will be exercise. If you, for example, choose to do a different exercise, like for example, you vary exercises rather than the duration, then you will keep the exercise variable and you change the duration. So the constant variable here instead will be the duration, and the independent variable will be the exercise itself. Okay, now with that, we can also think about something that can affect a runner or someone who wants to do an exercise. So think and try to put yourself in the shoes of the participants. If you were a participant, what could potentially affect your performance? For example, temperature could really affect how you run, for example. So if, for example, it was very humid or the temperature was high, then you won't be able to concentrate or perform as much. So these two actually are very important that we should keep them constant. So environmental temperature and humidity. Now, of course, the more the merrier here. Whatever comes in mind that you think is valuable, you should write because the constant variable can contribute to up to 3 marks from 6. So this part here alone can contribute to half of your grade for this specific question. Now, beside temperature and humidity, what you should also keep in consideration is, for example, the mass of the participants. Of course, the greater the mass, then the harder is it to do an exercise, to run, for example. So, mass of participants. Of course, the same thing goes with, like, of course, the age. Now, if someone was really too young, like for example 4 years old, then they won't have as much stamina. So this can affect the result. So you make sure that the ages are constant. So ages of participants. Okay. So with that done, we can move on into the method, which is basically the steps that you take to do the experiment. Okay. So first of all, what we want is that, for example, let's say that we are experimenting for running. Okay. So in this case, we will put the exercise here as running. Okay. So running with various durations. This is what we chose. So for running here, of course, we need what? We need treadmills. And you need to actually explain how you keep the environmental temperature constant, how you keep the humidity constant, and how you assess the participants themselves. So basically here, you gather participants and ensure they are of comparable age. Basically, the method you try to structure it like a procedure book. You go ahead, like basically you try to give this to someone who does not know how to do the experiment. So they will be able to follow through these steps and be able to essentially follow and do exactly what's written. So they're able to successfully do this experiment. So after you gather the participants, then you use a scale to see their mass. So you use a scale to assess participants' mass. Okay, now we need a way to keep the temperature constant in the room, which is by using an air conditioner, for example. So use an air conditioner with a fixed temperature. to ensure that the whole room has the same temperature, fixed temperature. And the same air conditioner can actually absorb and kind of like keep the humidity constant. So we can do that here. So use an air conditioner with a fixed temperature and humidity. So that is actually a 2-in-1. Okay, now beside that, we actually want to assess, for example, the speed of the participants on the treadmill. So actually, this is actually another way. So here, since we are actually doing running on a treadmill, then a constant variable that we can add here is the speed on the treadmill. So speed on the treadmill. Now you actually can already tell that this question has an insane amount of different approaches that you can take. So this is essentially what I'll be writing here. Use treadmills of fixed speeds and incline, and then run the experiment, let's say for 10 minutes. Or you don't really need to say 10 minutes, but you need to actually say that you should do it for a variable or a time period. So run the experiment for a time period. And this is actually where you should mention how you actually assess the time period using a stopwatch, of course. So for a time period, and then you count or you record using a stopwatch. So record time using stopwatch. Okay. Now after you're done with that, here, what you should be doing here, and this is actually a very, very important point. This could arguably be the most important point. You run the same experiment at the same speed, or actually at the same time, like the same duration, for about 3 times, and then you take the mean. So run the same experiment for the same duration at least 3 times, and then take mean. This is to ensure that like any random error would be eliminated. Not eliminated, but at least reduced. So run experiment, at least 3 times, and take mean. And then last but not least here, the last step here, is that you redo the experiment again. You run the experiment from scratch, but you do it with different durations. So here I said run the experiment at least 3 times. This is wrong. You should try 3 times at the same duration, or like for the same duration. You have to be specific for the same duration and take mean. And then for point 6, as we said, you run the experiment at different duration and different running duration. For example, let's say that in the first run, they did 10 minutes. So the second time you do the experiment, then you do it for like 20 minutes, for example. Now for safety and precaution, since this one doesn't really have the classic use a lab coat or wear gloves or wear goggles, then instead you have to be a little more creative and think about the well-being of the participants. So, since this is after all assessing the heart rate, you have to ensure that the participants do not have any past health record, which means they don't have any diseases or whatever. So this is actually a step that we should write here. So ensure participants don't have a health record or a previous health record. or disease record. So that's essentially what we should be writing here. Now also for the last point here, and I like to actually just underline everything, although it's not so easy digitally, but it'll be more clear in your exam. Like you underline the method, the safety and precautions and whatever, and it kind of like pops out more. So basically for the safety and precaution, the last bit here, you can also mention that there should be, for example, a medical team in the experimental area. So, like in the gym, for example, there was the treadmills that you are renting. There should be a medical team in case someone fainted or whatever, that they can intervene. So this is also a good step. Okay, so we're done with question number 2. That was a long one. And we're into question number 3 right now. Okay. Alright, so now here we have question number 3. Figure 3.1 is a photograph of a necklace starfish. Draw a large diagram of the necklace starfish shown in figure 3.1. Now, any drawing that you do here, it should be bigger than the figure here. So from P to Q, whatever that is, you ensure that basically your drawing is greater and bigger than this. That's your aim. Also, whatever outline you have, it should be constant and smooth. You have to ensure that it doesn't have something like so, where there's a huge gap in here. Or it doesn't have too much excess like so. Like in X crossing, you shouldn't have that. It should be one clear outline that looks something like this. Just give me a second. Something like this. Where it wouldn't exactly be clear where is the starting and the ending point. Now this one, I admit, may not be the best and prettiest drawing in the world. However, the idea stands. So we can apply the same principle here, and you do a blend drawing, which means you don't really draw the circles, because that would be very inaccurate if you do so. Instead, you just draw the overall structure and shape. So you try to imitate the shape. So let's try. I will try it like so. Of course, it won't be as easy drawing it on a digital tablet compared to, like, let's say, pencil. Now, some students would say that like the otherwise would be correct. However, personally, I would recommend, or I would always choose a pencil rather than drawing digital. Okay, so this is a drawing, for example, here. You try to draw it as accurately as possible, like so. Okay, now you can see here that there's a small excess section. This is actually wrong, so you shouldn't have this. So in your actual drawing, you should be able to just erase this excess bit, but since after all I'm doing it digital, I can't really. And you have to make sure that you do not shade. So the drawing that you do, you shouldn't just like draw it like so. Although after all, of course, it'd be easier to draw it shading, like using the shading method. However, this wouldn't be accurate. Why? Because you weren't really sure about how the outline would look like. Okay, so you should make sure that it is one continuous outline and drawing like so. Alright, so I went ahead and redrawn the drawing that it's closer to the actual shape. Okay, so now we have to make sure that we draw some details. So you can see that there seems to be something closer to what? To like a secondary outline, you can see here. Like there's a line of cells, like so, that acts like a secondary outline. So we should also draw the same thing here. Now, actually, the way that I'll be doing this, of course, is that I'll just be copying and pasting it. However, of course, you can do that in the exam. So you should also draw a secondary outline, like so, to represent what? To represent the secondary layer of the cells that will surround the main layer. Of course, in your actual exam, you should be using a thinner outline than the one I'm using. So what I can actually be using now is that I went ahead and redrawn the diagram so that it's closer to the actual shape. So you can see here that we actually have a secondary outline here, right? You can see that we have a secondary outline that represents a second wall of cells. Now you have a first outline here that just has the overall shape, and then you have a secondary outline for this wall of cells. So you can represent this wall of cells using just a secondary outline. So this is what I'll be using here. Of course, you should opt for a thinner pencil in your exam. This is not the ideal way to draw it. So you just draw the secondary outline. This is, of course, to represent the secondary wall of cells. Okay. Okay. Now, also, if you notice here in the drawing that I will come back in a bit after I'm done with this sketching here, or this drawing, is that essentially you would see that there is also a circle with a darker center. Now, you do not shade whatsoever in your exam. This is a no-no. You do not shade. So what you should do is that you only draw the circle in the middle and call it a day. That's it. So you draw the circle here, like so. And I will redraw that because the outline wasn't so clean. So you want to make sure that the outline does not touch like this. It should be a clean circle, not a perfect circle, of course, because that would be impossible. However, you try to make it so that it doesn't have an excess in the outline. Like it should match up pretty nicely here. Okay, that is good. However, you also notice here that there's also darker sections towards here with lighter tips at the end. So you can also represent that by just like a circle here to represent that section here. So you can do that here as well. Now, of course, this also includes the outer layer. So you can just draw that part at each step, like so. Because after all, this is only a plan drawing, not actually a detailed drawing. A detailed drawing, you will not be having that in your exam. You'll only be doing a plan drawing in which you only draw the outline and so and so. So you don't really have to worry about shading because it won't be accurate. The only way to have shading accurate is to look directly into the actual drawing. So this actually is satisfactory. You should not draw any cells like so. You should not do that. You should only be drawing the plan like this. That should be good. Okay, and you make sure that it is about the same size or bigger. So here, this is roughly the same size. This is borderline okay. However, it'd be better if it was bigger, like this, for example. Okay, so since I am happy with this result over here, except maybe I skipped drawing the circles over here, so I'll just be drawing that quickly. And then we can move on into the next section of the question. Okay, so with that clear, we can move on here. Line BQ in figure 3.1 represents the diameter of the nucleus starfish. Okay, measure the length of the line BQ in figure 3.1. Length of line BQ, okay, and then calculate the actual diameter of the nucleus starfish using the formula and your measurement. Give your answer to two significant figures. Okay, that's perfect. So essentially here, this line BQ, you use a ruler and then you measure it. Again, since I'm doing it digital, I can't really just use a ruler. So in your actual exam, you should be using your ruler here, your transparent ruler, and figure out this length from P to Q. According to the marking scheme, this length over here should be 93 plus 1, plus minus 1 millimeter. So 92, 93, 94 are accepted answers. So I will write here 93. And then calculate the actual diameter of the nucleus. Okay, so actual here, so this is actually the formula. Actual, if you rearrange it, would equal to the length over the magnification. Now the length we said was 93. The magnification here is 0.32. Okay, so essentially you just divide that. You have 93, divide by 0.32, and then you get 2.906. However, The question explicitly said that you should give your answer to two significant figures. So we'll only take this part here, which is 2.9. So our answer is 2.9 millimeters. This is the actual. Okay, now figure 3.2 is a photograph of a spiny starfish. Figure 3.3 is a photograph of a necklace starfish, which is exactly what we just drew. So the magnifications of the photographs are not the same. State two ways in the spiny starfish in figure 3.2 that differs from the necklace starfish in figure 3.3. Okay, what you can see here is that although both of them have circles in the center, the shading or the color is different. So here this one has a lighter color while this one has a darker color. So essentially, what the answer here should be, it should be that we can say that for the necklace starfish, the inner circle is darker, while the spiny starfish, this inner circle was light. So the necklace starfish featured an inner circle with a dark color compared to that of the spiny starfish. So we can say compared to the lighter tone of the spiny starfish. Okay, and this is actually a good opportunity to talk about the similarity. Because we already said that both of them have the same, or like have a circle in the beginning here, or like in the center. So we can say that both feature a circle in the center. Now for the second difference, you can also see and say that the spiny starfish has a more regular shape compared to that of the necklace starfish. And there are like circles here, and we'll discuss the name in a minute, in which they are present in the spiny starfish but not in the necklace starfish. You can also talk about the length, in which the length here is more uniform compared to the necklace starfish. So you can say about anything actually, but I will just use the easier one, which is that they both have, like the spiny starfish has a more regular shape compared to the necklace starfish. So, spiny starfish has a more regular shape compared to the necklace starfish. And we are done with this one. Okay. Now, if you're wondering about this second point here that we talked about earlier, which is like these circles here, these are actually called the ossicles. So you can say that the spiny starfish has ossicles, while the necklace starfish does not. Okay, we don't have much left, so let's move on. Alright, so here we have part B. Seven-armed starfish live in the sea. Scientists investigated the distribution of seven-armed starfish in one region. Now, what we were drawing here and comparing was a five-starred starfish. So this question is about a seven-starred starfish. Photographs of 20 meters squared areas of seabed were taken at different depths. Okay. The depth of the water in each area was measured when the water was at its highest level. The numbers of seven-armed starfish in each photograph was counted, and all photographs were taken on the same day of July. State two ways that the method used by the scientists ensured, within ED, that the numbers of seven-armed starfish counted at different depths would be compared or could be compared. Basically, the question is saying What part of this procedure ensures that the data are reliable enough for a valid comparison? So essentially, here we kind of need to tell the examiner what are the constant variables that were kept in consideration. For example, we can see here that all photographs were taken on the same day in July, which means that all the photographs were taken in the same season. So the season is constant. So all photographs are taken in the same season. And also you can see that there are standardized area for the photographs. So photographs of 20 meters squared areas of seabed were taken at different depths. So the area of photograph which was 20 meters squared was standardized. Okay, so that should be it. Now the results of the investigation are shown in table 3.1. Plot a histogram on the grid of the table in table 3.1. Okay, so basically we need to translate this into a visual image by drawing. Okay, so this is the histogram. We can see here, okay, so that's from 0 to 9.9 and then 10 to 19.9 or whatever. So really the last point is actually 50. Because when we draw the histogram, like the first example, let's say that's 10. So basically it would look something like this for, I'll just make that a bit thicker. Something like this, for example. And then it goes like so. So basically, this part here goes from 0 to 9.9 technically, and then it starts at 10 where it goes up. So when it ends, that's at the same idea, kind of, in which the line ends at 50. So what we should be doing here is that we should do a valid scale first. So we should end at 50. So if we start at 0 here, let's see if we can do 10, 20, and 30, and so on. So 0, 10, 20, 30, 40, and 50. So it's good. We can do like intervals of 10. And this will be here the x-axis in which we should label depth with the unit. So depth with the unit being milli or just meter itself. Okay. Now, how do you know which one should be the x-axis and which one should be the y-axis? Now, typically the x-axis is, in the case of histogram, it is for the range here. So this range will be the x-axis, and this here will be the y-axis. Or, if you want to see it in a more scientific way, then essentially the independent variable will be the x-axis, and the dependent variable will be the y-axis. Okay, so let's see, from 0 to 9.9, or technically 0 to 10, that will be 337. You can see that the greatest number is 1486. We can round it to 1500. So let's see what are the intervals that we can do. First of all, this table here, we don't really start at 0, so we don't need to start at 0. We can start at 100, for example. So here, that's 1, 2, 3, 4, 5, 6, 7, 8, and 9. Okay. So if we do by hundreds, in which, like, for example, this will be 100, then 200, and so on, we'll end up with 1000, which is nowhere near this. So we have to go with a different approach. Let's try intervals of 150, for example. So since here, this is less than 150, we can start at 0 here. So this will be 0, 150, 300, 450, 600, 750, 900, 1050, and then 1200. Again, you can see that it is not really up to 1500. So instead, the most valid way that we can do it is that we can do intervals of 200. So you have here 0, then you have 200, 400, 600, 800, 1000, 1200, 1400, and then 1600, in which the 1500 will be about here. Okay, that's good. And then we need to label, in which this will be the number of 7 armed starfish counted. You have to write that word for word. That will be the number of 7 armed starfish counted. Okay, now as we're done with the scaling part, we can actually plot. So let's go on. From 0 to 10, that'll be 337. Okay. Alright, so if we want to look at the intervals here, you can see here that we have 2 boxes, and we have an interval of 200, which would mean that each box here would equal to 20. So let's see, for example, if this one here was 100, because after all it's the midpoint, this would be 120, 140, 160, and 180. And this one here is 120. So it's better if we actually start with the least, for example, except you have an idea. So if we want to have 120, then it would be like so. But of course, this is not the correct range. They're just for like illustration. So this is how it would look like. Since it's not as easy to actually draw the line like so, now you can draw like just right over if you want to be more accurate. However, it won't really matter much because in the marking scheme, it is correct to have a box. So within your half a box range of the actual value, then you're good to go. So from 0 to 10, that's 337, which is closer to 340. So let's see what we have here. This is 300 over here. This is 320 and 340. So what I'll be doing is that I'll be drawing the line right under 340. Like so. If you've drawn it in the middle, it's also good. As long as you draw from this region here, like within this region, from the top to here, that'll be good to go. You're good to go. So I'll just draw like so. It'll make it a little bit thicker, so it's more visible. And then we can proceed with the next point here. So we have 1486. Okay. So that will be here, this part here. So 1400, 420, 440, 460, and 480. Okay. Now this here will be 490. So it's just around here. So you draw like so. Okay, so we're done with that. And then of course you have to connect them both like so. You have to connect them both. So you draw a vertical line. Okay. And then you have 20 to 29.9, which is like 20 to 30. So you have 1243. Okay, so that's 1200, 220, 240. So I'll just draw it around here. Then again, you connect the lines. Okay. And then here you have from 30 to 40, that will be 240. So this is 200, 220, 240. Since 41 isn't too different from 40, I'm just going to draw it in the same line. And then I'm going to connect. Okay. Just draw it more accurately. Okay, that sounds good. And then we can do the last one, which is being 121, which is just like 120. So that's 100 and that's 120, like so. Then you go ahead and, as you can tell, just connect the lines. And then do the same thing at last, and you're good to go. That's it. This is how you draw the histogram. This is how it should look like. All right, so I think we're done with it here. So for part 3 here, state the depth or the depth range in table 3.1 at which most 7-armed starfish were found, which is the one with the highest number, which is of course 10 to 19.9. So 10 to 19.9, just going to make that a bit thinner. 10 to 19.9. Okay, good. Now for the last question, using the information in table 3.1, calculate the total number of 7-armed starfish that were photographed at all depths. Okay. And then use this value to calculate the percentage starfish that were photographed in the range of 0 to 9.9. Express the percentage as a whole number, and then you have a space for working here. Okay. So they want it for all of the depths, right? Okay. So essentially, we need to go back into here, and essentially we want the total number of all of the depths, right? So essentially you have to just simply add them up. So you have, I'll just write that here and then copy paste it there. So you have 337 plus 1486 plus 1243 plus 241 plus 121. Okay. So essentially, this one here, that is what we want to do for our first step. Okay, I'm just going to just move that down. So essentially, this is the very first step that we do. We just add them up, we put them in the calculator, and then we get our answer. But we're not done yet. So let's first of all key that in. So 337 plus 1486 plus 1243 plus 241 plus 121. That would be 3428. Okay, and yes, you have to show the working, so be careful. So that is 3428. This is for the first part. But for the percentage of 7-armed starfish photographed at 0 to 9.9, essentially what you have to do is that you have to find the fraction of 337 compared to the total. And the way you do that is just by dividing them simply. So 337 over 3428. And to get a percentage, you just simply multiply by 100. So if you actually do that, that would be 337 divided by 3428. Multiply that by 100. You get 9.83. However, it says in the question, express the percentage as a whole number, which means that you round it to 10%. And that will be our answer. And that's that. We are done with this paper. Now this paper, I'd say that it's a bit lengthy compared to the ordinary. So I'd say that if you had any difficulties with this paper, you should do it again after viewing this video. You should do it again and then see if you're comfortable and you're okay with all the things that you initially got wrong. Because this paper was quite a good practice, I'd say. And this is a recent paper too. So that's that for today. We'll catch you in the next one. Goodbye.